Good morning, class 11th. Yes, today we will study about the ETS system or flow chart of the system, about the electron transport and also about the amphibolic pathway. So children, ETS system, electron transport system. So electron transport system, it is a chain of carrier molecules consisting of so many carrier molecules are there, just as NAD, nicotinamide adenine, then FAD or FMN, nucleotide, that is flavin mononucleotide if it is FMN, and if it is FAD then flavin adenosine dinucleotide. Then CO, coenzyme Q, this is also a carrier. Then some pigments are also used here about the cytochrome. See here, cytochrome B, cytochrome C, cytochrome A, cytochrome A3, that means type of A. It is cytochrome A3. This type of cytochrome pigments are also used, in which are present in mitochondria, and in the mitochondria there are F1 particle. So in that F1 particle it goes on. This is the ETS system or electron transport system. So after glycolysis, the pyruvic acid will enter to the Krebs cycle in presence of that coenzyme A, acetyl coenzyme A. See, we will start the ETS just after the Krebs cycle. So Krebs cycle, coenzyme A, this is coenzyme A, it is having hydrogen atom with it, so it will oxidize and release the coenzyme A because it acts as the catalyst and it releases hydrogen atom. Then this hydrogen is taken by the NAD+. NAD+ will collect or will attach or will combine with the hydrogen atom and release one hydrogen atom. Now next, when there will be the formation of NADH+, and again this hydrogen atom, it is released and again NAD+ will be released to continue the cycle again and again, that is reduction and oxidation. And this hydrogen, when it is released, it is carried or taken by the FADH. FADH, it collects or it combines with the proper hydrogen molecule. So FAD will form the FADH2. When FADH2 will form, again it will release hydrogen molecule and it will release the FAD free, because again it will continue the next oxidation and reduction process. Again it will carry the hydrogen and again hydrogen will be given to the another compound and again FAD will be free. This hydrogen atom, hydrogen ion molecule is taken by the coenzyme Q. Still you know about the coenzyme A. In that ETS system there is the coenzyme Q. Coenzyme Q will collect that hydrogen atom, hydrogen molecule, and will form coenzyme QH2. Then after again see, it releases the hydrogen atoms, two hydrogen atoms from one molecule of hydrogen, and it again makes free the coenzyme Q to complete the next cycles. Then Here 2 hydrogen atoms are free. Now this Fe3, 3 means Fe plus 3, it means the ferric. So whatever iron we have in our body or plant's body, ferric compound will be changes into ferrous. So children, ferric will be changes into ferrous, that means plus 3 will be converted into plus 2 according to charges. If the charges will be less, it means some electron will be there to complete that orbit. So here ferric will be changes into ferrous in presence of cytochrome pigment B. Now in the next step, again see Fe2 becomes Fe3, that means again it will lose electron, and again it come to the next step in presence of cytochrome C pigment. Cytochrome C pigment, again see it was Fe3 will be free, and then Fe2 again in presence of cytochrome C again will lose the electron and will form the Fe3, that means from ferrous to ferric. Here from ferric to ferrous, and here from ferrous to ferric. Then see, I made here one symbol here. From here come to the next place here. Then again ferric will be changes into, see, ferric will be changes into ferrous, ferrous will be changes into ferric, again ferric will be changes into ferrous in presence of cytochrome A pigment. All these are present in the F1 particle of the mitochondria. So in presence of cytochrome A, ferric will be changes into ferrous, and continuous flow of electron will goes on from a higher to a lower energy level. That's why it loses some energy also. So here there is the formation of ATP along with the inorganic phosphorus and ADP, which energy is consumed already, that is ATP will be already changes into ADP. It will collect that inorganic phosphorus, forms the ATP. Here also again ADP collect the inorganic phosphorus and will be converted into ATP. In this way, in the total series, they are continuously giving some energy to continue the system or flowchart of it. Then according to here again ADP will collect inorganic phosphorus and will form again the energy giving molecule, that is ATP. Then next to it, see, ferrous will be changes into ferric, again it will lose one electron, and that electron in presence of cytochrome A3, this is the part of cytochrome A pigment, it is part that as we have some shades of the color, yet it is not proper red but a little bit this shade and this type of shade is here, about the red only and about the blue only. So color will not change but shades are changed, different. So in this way here the pigment will be the same, cytochrome A, but the type of the cytochrome A pigment is A3. Now in presence of A3, that ferric, ये देखिए ferric बना था यहाँ पर, so ferric it was here, so ferric again will be changes into ferrous. Now children, 2 hydrogen atoms were released here. These hydrogen atoms and half of the oxygen molecule, it means one oxygen atom. These two will combine and will form the water. So this is the proper flow chart of the electron transport system. And here one coenzyme Q, you have to use it, that means plants are using it rather than the coenzyme A, because coenzyme A only it was used in the Krebs cycle. Then it is passed along the cytochrome pigments, and after the cytochrome pigments, the reactions takes place, and at the last it will form water molecule. And 3 ATP molecules are formed, which are some amount they use in their to get their energy, and some are releasing there in that ETS system. Now children, amphibolic pathway. As children, according to chemistry, you studied about that amphoteric oxides or amphoteric elements, those who can react with the acids and bases both together. So children, it is known as amphibolic pathway because the name is given. It contains or it consists or it is having both the processes which is catabolic and anabolic. Both types of activities are going on in this pathway. So this is known as the amphibolic pathway. Amphi means both. As children, you studied about the amphibia. Those animals can live in two types of habitats. So they are known as amphibians. So here amphi word is given for the both catabolic and anabolic processes. Now children, in class 10th you studied about the hormones. In case of pancreas, there is one process, that is gluconeogenesis. So children, gluconeogenesis is the process in which our diet, that means maybe it is carbohydrate, maybe it is protein, maybe it is fat, from all type of nutritions, they extract the glucose molecule and it goes for our respiration, and it help to us to release some energy. So this is the amphibolic pathway in which all types of nutrient can be changed or converted into glucose, and this glucose will be oxidized and will give us energy. So there are some formation of few compounds. So this is an anabolic process also, and there is the destruction of glucose in presence of oxygen. So it is catabolic also. So in this way, this is called as amphibolic pathway. So children, we will start from here. See, when our respiration starts, first process is glycolysis. So glucose, that is 6-carbon compound, will be changes into triose sugar. Triose sugar, that means the compound will be formed with the 3-carbon. So these are known as triose sugar. As children, you studied in glycolysis that PGL, phosphoglyceraldehyde, and trihydroxyacetone. These are the two compounds which are the triose sugar. Then this triose sugar will continue the full process of glycolysis, and lastly there will be the formation of pyruvic acid. So here it is pyruvate. Then after the formation of pyruvic acid, before entering to Krebs cycle, it will form nearly about the 4 steps are there. It will form another compound, it is acetyl coenzyme A. With the help of catalyst coenzyme A, it will form acetyl coenzyme A. These 4 steps are very essential to enter the Krebs cycle. Now this acetyl coenzyme A will release carbon dioxide, whatever, as per Krebs cycle. It will form citric acid. Children, all the steps I am not writing here because you also don't have to write, but in a simple way, because it is citric acid cycle. So please, you have to write the citric acid formation. It will form 6-carbon compound. It is citric acid. Then citric acid will continue the processes found in Krebs cycle, and it will form alpha ketoglutarate acid. alpha ketoglutaric acid is 5 carbon compound, and then alpha ketoglutaric acid at the last it will form oxaloacetic acid, and this oxaloacetic acid by taking water molecule again it will form, in presence of acetyl coenzyme again it will form citric acid. So this is the repetition of the same cycle, that is Krebs cycle. And now when we have taken the fat will be digested into glycerol and fatty acid. So when this glycerol has to go in our respiratory tract or respiratory process, then this glycerol in presence of phosphoglyceraldehyde PGAL, it will be changes this glycerol into phosphoglyceraldehyde, and then the reaction will goes on. It will form the pyruvic acid, and remaining, that is fatty acid. Fatty acid, it does not enter to the glycolysis. It enters in the acetyl coenzyme A. After the formation of acetyl coenzyme A, this fatty acid will be here, and it can go along with the Krebs cycle. So again here it will give the facility to continue the process in the Krebs cycle. So this is fatty acid along with the acetyl coenzyme A, and will continue the same process as follows in the Krebs cycle. It will form citric acid, then alpha ketoglutaric acid, and then oxaloacetic acid. Again oxaloacetic acid will leave the acetyl coenzyme A and will form the citric acid. If suppose we have taken the protein, then protein will be digested in the along with the peptidase and peptides it will form, and after that it will be fully digested and blood will absorb that amino acid. This amino acid again will be included with the pyruvic acid, not in the glycolysis pathway. After finishing the glycolysis pathway, it will be included with the pyruvic acid, and it will also continue with the formation of acetyl coenzyme A, and again it will continue along with the Krebs cycle. So if it is totally digested, then it is amino acid, and amino acid will go to the pyruvic acid. Or this amino acid, if it does not combine with the pyruvic acid or does not included with the pyruvic acid, it will directly come to the alpha ketoglutaric acid. And alpha ketoglutaric acid, when it is included here, again it will go along with the Krebs cycles processes, malic acid and other acids, then it will form oxaloacetic acid. Oxaloacetic acid, again acid with the acetyl coenzyme A, it will form the citric acid. So this is the this is the proper formation of our respiratory process, by which we can understand the types of activities, either it is catabolic or it is anabolic, but both are continuously going on in the same process. So this is named as the amphibolic pathway, and this is the reason why we are not eating suppose carbohydrate, then also we are aspiring throughout the day, throughout the night. If we are not taken protein, then also we are aspiring. Or suppose we are not getting any food, our stomach is empty, then also we are aspiring. Because of that, glucagon, that stored material, will be changed into, that is glycogen will be converted into glucose, and our respiration process will goes on. So these are the processes by which we can continue our respiration, whether we have not taken carbohydrate, whether we have not taken protein, whether we have not taken fat. So these are the processes. Okay children, thank you. God bless you.